Hello students, welcome to Read MedPrep Academy channel. Today in chromosomal basis of inheritance part 2, we are going to talk about linkage, coupling and repulsion theory, kinds of linkage and linkage groups. First, let us move on to linkage. The genes which determine the character of an individual are carried by the chromosomes. The genes for different characters may be present in the same chromosome or in a different chromosome. When the genes are present in the different chromosomes, they assort independently according to the Mendel's law of independent assortment. We have already seen in our previous classes the dihybrid cross segregation of gametes by the law of independent assortment. Let us take the round seeds with yellow cotyledon, which is homozygous parent, with the genotype capital R, capital R, capital Y, capital Y, and a homozygous character which is wrinkled seeds with green cotyledon. The parental genotype is small r, small r, small y, which is recessive. And the gametes formed by capital R, capital R, capital Y is capital RY. And the gametes formed by small r, small r, small y is small r, small y. And in the F1 generation, the progeny obtained is heterozygous. The genotype is capital R, capital Y, small y. When they are selfed, the round seeds and yellow cotyledon will produce four types of gametes: capital R, capital Y, capital R, small r, capital Y, and small y. The genes are present on separate chromosomes and random assortment takes place. So, four different types of gametes are formed in equal proportions. This obeys the law of independent assortment. Biologists came across certain genetic characteristics that did not assort out independently in other organisms after Mendel's work. One such case was reported in sweet peas, Lathyrus odoratus, by William Bateson and Reginald C. Punnett in 1906. They crossed one homozygous strain of sweet peas having purple flowers and long pollen grains with another homozygous strain having red flowers and round pollen grains. All the F1 progenies had purple flower and long pollen grains, indicating purple flower and long pollen as a dominant character represented as capital P, capital L, capital P, capital L. Over red flower, round pollen, small p, small l, small p, small l. When they crossed the F1 generation with the double recessive parent as a test cross, the results in F2 progenies did not exhibit the 1:1:1:1 ratio as expected according to the law of independent assortment. A greater number of F2 plants had purple flowers and long pollen or red flowers and round pollen. So they concluded that the genes for purple color and long pollen grain and genes for red color and round pollen grain were found close together in the same homologous pair of chromosomes. What is linkage? These genes do not allow themselves to be separated, so they do not assort independently. This type of tendency of the genes to stay together during separation of the chromosomes is called linkage. Genes located close together on the same chromosome and inherited together are called linked genes. But the two genes that sufficiently far apart on the same chromosome are called unlinked genes or syntenic genes. Here in this image you can see the linked and the unlinked genes on the chromosome. On the left side you can see that the gene 1 and gene 2 are present so close together in the lower arm of one chromosome. So these genes are linked genes. In the middle you can see that gene 1 is present in the upper arm of a homologous chromosome and gene 2 is present in the lower arm of another homologous chromosome. They usually are not inherited together, so they are unlinked genes. On the right side you can see that in the lower arm of one chromosome there is gene 1 which is far apart with that of the gene 2, and these two genes are unlinked genes. They are not inherited together. Now what is cis-trans arrangement of genes? Here you can see on the left side the cis arrangement, where one homologous chromosome has a dominant allele of one character and another dominant allele of another character. whereas in another homologous chromosome, there is a recessive allele of one trait and the recessive allele of another trait. This is a cis arrangement of genes. What is trans arrangement of genes? Where capital A small b and small a capital B is seen, where in one homologous chromosome the allele of a dominant character is present and the recessive allele of another character is present. In another homologous chromosome, the recessive allele of one trait is present and the dominant allele of another trait is present. So, cis arrangement of genes, the presence of dominant alleles of two or more pairs on one chromosome and the recessive alleles on the homologous chromosome. What is transconfiguration? Trans arrangement of genes where an arrangement in which the dominant allele of one pair of genes and the recessive allele of another pair of genes are present on the same chromosome. Now, what is this recombination frequency? It is to be differentiated by the value of recombination frequency. If the recombination frequency value is more than 50%, the two genes show unlinked. When the recombination frequency value is less than 50%, they show linked genes. Closely located genes show strong linkage, while genes which are far apart, which are located far away, show weak linkages. Now, let us move on to coupling and repulsion theory. What is coupling or cis configuration? The two dominant alleles or recessive alleles occur in the same homologous chromosomes. They tend to inherit together into the same gamete, and this is called coupling or cis configuration. Here you can see in this image, the purple flower with long pollen is taken as one parent and the red flower with round pollen is taken as another parent. The crosses are made between the homozygous parents. The purple flower with long pollen will have capital P, capital L in one homologous chromosome and capital P, capital L in another homologous chromosome. Whereas when you take the red flower with round pollen, the small p and small l is taken in one homologous chromosome and the another homologous chromosome contains small p and small l. The gametes formed by the purple flower long pollen is capital P, capital L, and the gamete formed by the red flower and round pollen is small p, small l. And the F1 generation that is formed has purple flower with long pollen. The genotype is capital P, capital L and small p and small l, where capital P and capital L is present in one homologous chromosome and small p and small l is present in another homologous chromosome. When a test cross is done, where the heterozygous F1 crosses with the double recessive parent, the purple flower with long pollen, which is represented as capital P, capital L in one homologous chromosome and small p and small l in another homologous chromosome, is crossed with red flower with round pollen, which is small p and small l in one homologous chromosome and small p and small l in another homologous chromosome. The F1 generation will produce gametes. So there is less chance of crossing over due to the linked genes. And the gametes produced by purple flower long pollen is capital P, capital L, capital P, small p, capital L and small p, small l. Gamete that is produced by the red flower and round pollen is small p and small l. In the F2 generation that is formed, it has purple long flowers, purple round, red long and red round. The purple long is represented as capital P, capital L in one homologous chromosome and small p and small l in another homologous chromosome. In purple round, it has capital P and small l in one homologous chromosome and small p and small l in another homologous chromosome. In red long, capital P and capital L in one homologous chromosome and small p and small l in another homologous chromosome. In red round, small p, small l in one homologous chromosome and small p and small l in another homologous chromosome. So with linked genes, non-recombinant progenies are predominant. So the parental progeny is purple long and red round, and the recombinant progeny is purple round and red long. The observed ratio that is obtained here is 44:6:6:6:44. That means 7:1:1:1:7. Normally, the expected ratio should be 1:1:1:1. Now, what is repulsion or transconfiguration? If dominant or recessive alleles are present on two different but homologous chromosomes, they inherit apart into different gametes are called repulsion or transconfiguration. Here you can see in this image the purple flower with round pollen and a red flower with long pollen, which are the parents, and the crosses between the homozygous parents is done, and the purple flower round pollen will have capital P small l in one homologous chromosome and capital P and small in another homologous chromosome, and the red flower with long pollen will have small p with capital in one homologous chromosome and small p and capital in another homologous chromosome. The gametes formed by the purple flower round pollen is capital P small l, and the gamete formed by the red flower and long pollen is small p and capital L. And the F1 progeny form is purple flower with long pollen. The genotype is capital P small l in one homologous chromosome and small p and capital L in another homologous chromosome. When a test cross is done between the F1 generation and the recessive trait, the heterozygous F1 crosses with the double recessive parent. The purple flower with long pollen is represented in one homologous chromosome as capital P and small l, and in another homologous chromosome small p and capital L. The red flower and round pollen will have small p small l in one homologous chromosome and small p and small l in another homologous chromosome. And the gametes formed because there is less chance of crossing over due to the linked genes. The purple flower with long pollen will produce capital P L, capital P small p capital L, and small p and small l. Whereas the red flower with the round pollen will produce small p and small l. So in the F1 generation, the progeny obtained is purple long, purple round, red long, and red round. The purple long will have capital P capital L in one homologous chromosome and capital P and small l in another homologous chromosome. The purple round will have capital P and small l in one homologous chromosome and small p and small l in another homologous chromosome. In the red long, small p and capital L will be present in one homologous chromosome and small p and capital L will be present in another homologous chromosome. In the red round, small p and small l and small p and small l are present in the homologous chromosomes. With linked genes, non-recombinant progenies are predominant. That means the purple long that is formed and the red round are recombinant progenies, whereas purple round and the red long are the parental progenies. So the ratio obtained is 6:44:44:6. This is the observed ratio, which is 1:7:7:1. Normally, the expected ratio should be 1:1:1:1:1. Now, let us move on to the kinds of linkage. T.H. Morgan found two types of linkages. They are complete linkage and incomplete linkage, depending upon the absence or presence of the new combination of the linked genes. If the chances of separation of the two linked genes are not possible, those genes always remain together. As a result, only parental combinations are observed. The linked genes are located very close together on the same chromosome. Such genes do not exhibit crossing over. This phenomenon is called complete linkage. It is a rare but has been seen in male Drosophila. C.B. Bridges in 1919 discovered that crossing over is completely absent in some species of male Drosophila. Here you can see in this image the parents producing gametes capital A, capital B, capital A, capital B in the homologous chromosomes, small a, small b, and small a, small b in another parent of the homologous chromosome. The gametes produced are capital A, capital B by one parent and small a, small b by another parent. The parental combination that is obtained is capital A, capital B and small a, small b. This is a complete linkage. Only the parental combination is obtained. In incomplete linkage, if two linked genes are sufficiently apart, the chances of their separation are possible. As a result, parental and non-parental combinations are observed. This phenomenon is called incomplete linkage. This was observed in maize. It was reported by Hutchinson. Here in this image you can see the parental combination having the chromosomes capital A, capital B in one homologous chromosome, capital A and small b in another homologous chromosome. In another parent, in one homologous chromosome it is small a, capital B, and small b. The gametes produced by one parent, capital A, capital B, and capital A small b is capital A, capital B, capital A small b. The another parent produces small a and capital B and small a and small b. Here, the non-parental combination is obtained the incomplete linkage, like having capital A, capital B and small a, small b. Now let us move on to the linkage groups. The groups of linearly arranged linked genes on a chromosome are called linkage groups. In any species, the number of linkage groups corresponds to the number of haploid set of chromosomes. The linkage groups in some organisms, Mucor has 2 linkage groups, Drosophila has 4, Sweet pea has 7, Neurospora has 7 and maize has 10. So linkage and crossing over are 2 separate processes that have opposite effects. Linkage keeps particular genes together, but crossing over mixes the genes together. So what are the differences between the linkage and the crossing over? In linkage, the genes present on the chromosome stay close together, whereas in crossing over, it leads to separation of linked genes. In linkage, it involves same chromosome of the homologous chromosome, whereas in crossing over, it involves exchange of segments between non-sister chromatids of the homologous chromosome. So linkage, it reduces the new gene combinations, whereas crossing over, it increases the variability by forming new gene combinations, leads to formation of new organism. So today, in chromosomal basis of inheritance part 2, we discussed about linkage, coupling and repulsion theory, kinds of linkage and linkage groups. Thank you. Kindly subscribe, like, share and comment our channel Read MedPrep Academy. Log on to www.readmedprepacademy.com for MCQs. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Thank you very much.